Should I start? Good afternoon, everyone. I, Dr. Neha Batra, on behalf of Agarwal College, Balragarh, welcome you all for the third day workshop on computer-aided drug design. By now, I am sure that everyone is well aware of the theme and purpose of this workshop. I personally am highly benefited by getting this opportunity of live, hands-on training on various available softwares. Not wasting any time further, I would take it as an extreme privilege to invite our honorable speaker, Professor Mukesh Doble, to conduct today's session. Over to you, sir.
Thank you very much. Hello, everyone. Welcome to today's workshop. I hope you did practice yesterday and understood how to develop a pharmacophore-based approach. And you must have used PyMol, which is a very useful software which can do a lot of graphical viewing. And then the other two, Pharmagist, Zinc Farmer, they are all online servers. Okay, so they are free for us to use actually. Today we will continue and we will go further. Okay. Can you see my screen?
Yes, sir.
Okay. So we have been looking at this left-hand side. That's called ligand-based design. Ligand is your lead molecule or hit or drug or whatever it is. That's a small molecule. On the right-hand side, we have the target. That is the protein or enzyme. The molecule goes and binds to that part. So that means we should have some idea about the target protein or enzyme. So in this left-hand side, we don't need to know anything about our target. Okay, so that's why this is much easier to do. We can combine both ligand-based and target-based also. So yesterday we saw the pharmacophore-based design. So we identified common features of a set of molecules, which we took from literature, maybe 4 or 5 molecules we took from literature, and see what are the common features like hydrophobic group, aromatic, hydrogen bond acceptors, donors, and charged, anions and cations. So once you identify these pharmacophores, we can go into other databases, for example, zinc database, and then see whether there are any molecules with that feature, pharmacophore feature. So hopefully they will also have similar activity, because all the molecules we selected originally had the activity. So when we identify new molecules with that feature, hopefully they will also have the activity. That's how we selected it. The software will select almost a lakh of molecules actually. Then you go through each one of them, check for their drug likeness property. We can use Swiss ADME. We must be very now thorough with Swiss ADME. We can use the ADMET tool, the toxicity tools, and see whether which ones satisfy the drug likeness property, which ones does not. So if it does not satisfy, we just park it. We don't spend much time. Those which satisfy are a possible lead, and they will also have the drug likeness property. That's why the entire logic of it. Today we will look at QSAR, quantitative structure activity relationship based approach. Here you are going to, because it says quantitative, so we are going to develop a relationship, mathematical relation between activity and the structural features. Okay, so there will be a mathematical relation. Whereas in pharmacophore, we never had mathematical. It just identifies common features. Whereas in QSAR, we will develop a mathematical relationship between activity, say on the left-hand side, and various structural features. It's also called structural descriptors on the right-hand side. Okay, so you must have all done, say, regression, y equal to mx plus c, right? So y is your dependent variable, that is activity. X is your independent variable. That could be a structural feature or structural descriptor. We can have y equal to m1 x1 plus m2 x2 plus c also, or we can have m1 x1 plus m2 x2 plus m3 x3 plus c, like that, you know. So each of these x are independent variables, or structural descriptor. I'll use descriptor here. So just like us, no, we all have descriptors, right? If I take me, height is one descriptor, weight is descriptor, length of my arm is a descriptor, length of my leg is a descriptor, length of my hair is a descriptor, even weight of my heart, that could be a descriptor, WBC count, RBC count, weight of my liver, like that, you know. I am going to have many, many descriptors. So for each human being also, we can have 2000, 3000 descriptors. Similarly, for molecules also, we can have 3000 descriptors like this. There are softwares which can calculate these descriptors. Okay, that is all the x's. X means your independent variable. We are trying, then we will try to get the best x, which will develop an equation with the y. Okay, that is what the quantitative structure activity relationship is all about. Okay. So basically, just like you selected the 4-5 compounds in Pharma 4 from literature, here we will select 4-5 compounds plus also their activity values, you know, experimental values, because here we are going to do a quantitative structure activity relation. So I will also need the activity numbers. No. So not only 4 or 5 molecules you select for a particular disease, but even their activities also you need to get, okay, from the literature. That's very, very important. Okay, that's very, very important. And then you calculate lots of descriptors using software, and then you select the best descriptor using some statistical techniques I will show you, and then you develop a mathematical relation between y equal to either mx plus c or m1 x1 plus m2 x2 plus c, like that actually. Okay, that's what the whole thing is. Okay, descriptors. What are these descriptors? There are many, many, many types of descriptors. Like I said, there are something called zero order descriptors, first order descriptors, so many types of descriptors. Okay, there are zero order descriptors like molecular weight, average molecular number of atoms, number of hydrogen atoms, number of carbon atoms, number of heteroatoms, double bonds, number of double bonds, number of triple bonds, aromatic bonds, rotatable bonds. They are called zero order descriptors. Then we have first order descriptors like number of primary carbon, that is sp3 carbon, secondary sp2 carbon, sp carbon, quaternary carbon, tertiary carbon in a ring, carbon in a ring, aromatic carbon, like that, you know. They are called one-dimensional descriptors. Topological descriptor. Topological means all related to graph theory. What is the distance between longest distance between one carbon and another carbon? What is the shortest distance? They are all coming from the graph theory, concept of graph theory. Okay, that's called topological descriptors. Then we have three-dimensional descriptors, radius of gyration, then van der Waal surface area, then polar surface area, solvent accessible area, hydrophilic surface area, volume of the molecule, electrostatic charged partial surface area, highest occupied molecular orbital, lowest unoccupied molecular orbital, potential energy difference between these two, minimum energy conformation, binding energy, ionization potential. All these are three-dimensional descriptors. Okay, and then you have descriptors like log p, then which cannot be called anything actually. So huge number of descriptors are there. Okay. Electrostatic, volume-based descriptor, graph theory-based descriptor, one-dimensional descriptor, zero-order descriptor, like that, huge number of descriptors. And then you make use of this to develop a mathematical relation with the y versus x. So there are different approaches are there. Hansch analysis is there, and then there is a free Wilson analysis. Then there are many statistical methods are there to develop a mathematical relation between the descriptor, that is the x, with the y. So in the Hansch analysis, what Hansch did was they said activity, that is y is equal to a plus say log p, that is one independent variable, plus b into some electrostatic factor, that is another descriptor, another independent variable, and c into steric effect. Okay, that's another descriptor, plus d. So you can have models like that. Not that every time you will have models like that, but you can think of models like that, or you can think of spire also like that. So basically, a QSAR is nothing but a regression relation activity on the left-hand side, which is the dependent variable, and M1x1 plus M2x2, M3x3, all the x1, x2, x3 or the independent variable or the descriptors. Okay, then you try to get the best A, B, C by fitting all the data. Suppose you have selected 5 data points and you have got 5 values, you get that. Okay, that's called the Hansch based analysis, and we will also be using that. There is another approach which is called free Wilson approach. What they do is activity, there is a basic number, and whenever you add a group, suppose I add OH group, activity may go up or go down. So this one is effect of each group which you are adding as a substituent to the parent molecule. Okay. It could be OH, it could be OCH3, it could be aryl group, it could be long chain hydrocarbon, like that. So effect of each one on the activity that gets added up. So you have a parent original structure, and then you are having derivatization, or different types of functional groups you're adding. So effect of each functional group on the activity. It may be going up, maybe going down also. Say maybe some electron withdrawing groups, the activity may go down. Electron donating groups activity may go up. So this A can be plus or minus. So this approach is based on group contribution method. That is each contribution of each group on the overall activity. Whereas this approach is more like a regression based approach. You must have been all familiar with regression based approach. So that is what we will be doing. Regression based approach. Okay. So you get some data from the literature with the activity value. So that will be the Y. Descriptors you calculate, descriptors or properties or features or whatever it is, and then you develop a regression relation. And depending upon the number of data points, you develop a regression relation. Okay, you develop a regression relation, so that you get a good fit. Okay, that's the QSAR equation. Okay, which is a equation like your regression relation. Now, what is the use of this? The use is, suppose I have a new molecule. I am designing a new molecule on the computer screen as a medicinal chemist. And before going to lab, you want to know whether that molecule will have good activity or bad activity. So I can calculate the descriptors for that molecule, substitute in the equation, and I can predict. Okay, I can predict what will be the activity of that molecule. Okay, in pharmacophore we can't predict activities. If you see that main difference between pharmacophore and QSAR, QSAR we can predict activity. In pharmacophore it just selected lots of molecules. Okay, using zinc pharma. Whereas in QSAR, you can predict the activity using that equation. That's the main advantage. That is why You must start with some set of molecules from literature with the activity data available from the literature. This is the entire concept of QSAR. The advantage is we can use it to predict activity of new molecules. So if I have a QSAR equation, I design many molecules using Swiss ADME. Okay, hundreds of molecules are designed. Now I can use this equation, QSAR equation, and try to predict activity and select only those which shows good activity, predicted activity. Rest of them you leave it, and then only take those and maybe go to my wet lab. Okay, that way we can use it as a tool for shortlisting candidates also. That's the main advantage. Okay? So many descriptors are there. Let's do an example. You can do QSAR, that is regression. You can use Excel itself. We can do linear regression, multiple linear regression. So many things we can do in Excel actually. I just will show you. Imagine I got some anti-cancer activity. I went to literature, collected 5 molecules whose activity is known in the literature. Okay, 5 molecules. I calculated 3 descriptors. Actually, you can calculate 3000 descriptors. For the first starting example, I will show you. 3 descriptors. This is volume of this molecule, log P of this molecule. This is dipole moment of this molecule. Okay. So similarly for molecular 2, this, 3, this, 4, this, 5, this. Okay. So I have 5 activities and descriptors. Now, with there is a rule which says for if you want to have one descriptor model, that is y equal to mx plus c, that is one descriptor, we should have at least approximately 5 data points. Okay, so if you want to have 2 descriptor model, that is y equal to m1 x1 plus m2 x2 plus c, then you should have approximately 10 data points. These come from statistics actually. If you have done course on statistics and regression, you will come across this. So in this problem, I can develop only one descriptor model, y equal to mx plus c, but I want to see which x is good, whether molecular volume is good, or log P is good, or dipole moment is good. I can nicely do it in Excel also. I'll show you first. Okay, so what I did was I put the data in the Excel. Okay, all the data I have put in, 2.34 activity, 1.89, 0.236, all these things. So you can see in Excel also. Can you see this? So 2.34, 1.89, 0.233, 0.6, then 420. This is volume 3, 32, 190, 467, 59, log P 2.8, 2.6 minus 3. Then dipole 0.97, 2.3, 3.3. Okay. Okay, first thing is I need to see which descriptors are good, describe the activity. And there is a command called correlation. Correlation is the relationship between the X and the Y, which descriptor. So Excel has a command CORREL, correl, correl. So you give this column and this column. Okay, this is the command for determining the correlation. Okay, 0.95. So closer to 1, correlation coefficient can change between -1 to +1. So closer to 1 means highly correlated. Closer to 0 means lowly correlated. Okay, so this is very good. So the correlation between volume and activity is very, very good actually. 0.95 is coming closer to 1. Especially when you are doing experimental studies, you will not get such big high correlation coefficient because there are a lot of experimental errors will be there, assay errors. Biology always gives a lot of errors. You may go up to 0.7. Okay, now let us do correlation between log P and I hope you are quite familiar with Excel. So the correlation between log P and activity is bad, only 0.38. So we cannot make a model with log P and activity because it's bad. We can definitely make a model with the volume versus activity because it's very, very good, very, very good, closer to 1. Now let's look at dipole. This is also very good. It's showing minus. Minus means as dipole increases, activity goes down. As dipole decreases, activity goes up. Whereas the case of volume, as the volume increases, activity goes up. Volume decreases, activity goes down. So this is also good. Even if it is minus, it's closer to -1. Can you see? Both are good. Volume, so we can either think of a QSAR with volume or with dipole. And as I said, we will do only one parameter QSAR because I have only 5 data points. The statistics always says that don't go beyond one parameter model. It's based on the degrees of freedom. You must have heard of degrees of freedom, p-value, all those things actually in statistics. So QSAR uses a lot of statistics. Okay, now let us plot it and see. First, volume versus I. So my x, that is the independent variable is volume. Y, my dependent variable is activity. I hope you all know how to draw graphs and all with the Excel. Okay. So you see, as the volume increases, this is the volume, x-axis is volume, y-axis is activity. So as the volume increases, activity increases. So it looks like a nice graph actually. So we can get the equation for that. We can first try to fit a straight line, add a trend line. It is called. So it gives you an equation. Okay, so this is good. R square, that is square of R, very high value, closer to 1. So this is a very good model. You can see the data, the line fitting the data. X-axis is your volume, y-axis is activity. So this is a QSAR. Activity equal to 0.0117 volume minus 2.0254. Okay, this is a QSAR model for this set of 5 data points actually, and it's a good model because R square is very good. Now, what is the use of the model? So suppose I have a thinking of design synthesizing some 10 compounds, different structures and all that. Okay, for the same anti-cancer activity, I can use Swiss ADME, calculate the volume, and then I can put the volume in the X place, and then it will predict what will be the activity. Okay, so we will know whether the activity is better than this or equal to this or worse than this. So those 10 compounds, we can see which ones look very good, promising, which one look bad. So only the promising I could take further for my synthetic in vitro studies actually. So that's the use of QSAR. Okay, so this is volume and the x-axis, and activity on the y-axis, and this is your QSAR. Activity equal to 0.0117 into volume minus 2.254. Okay, let us. I know we know log p is very bad because correlation coefficient is bad, but anyway we will try to see how it looks like. So log p in the x-axis and activity in the y-axis. I'm trying to plot. You can see it's all very bad. So it's going upper nature, upper nature. So even if you try to make an equation and trend line, you will see the points are. This is the equation, and points are all over the place. And R square is also very bad, as expected, because our correlation coefficient was bad. So log p is not a good model. You can see this. See this one, all the points are very close to the straight line. Here you see. So definitely this is not a good model. Correct. Now let us look at other one. The third one, dipole versus activity. Okay, so let us look at dipole versus activity. Dipole versus activity. X is your dipole, Y is your activity. No, this is also good. It's negative slope because your correlation coefficient came to be negative as expected, right? So we can even have this. This is also looking good. Trend line. So you see so activity equal to minus 1.0411 dipole plus 3.96. Here also R square is good, 0.88. But this is the best. R square is 0.92, but this also is reasonably good. It's showing a negative slope. So dipole increases, activity will go down. So if I have to design molecules, I should have lower dipoles and higher one. So this is the best QSAR we got. This is also a reasonably good QSAR. We can use it. If you want to use this QSAR, I should know dipole the molecules. If I have this is definitely not good. Log peak QSAR is very bad. Volume is good, right? So we can use. Excel to do this type. for a one parameter. We can even do two parameter model. Although with 5 data points, I said it is not good, but we will anyway try. In real life, please don't do that. So I will copy this. Ideally, you should have approximately 10 data points. So I will remove log P. So I will use only volume and dipole and see whether I can make a model. Y equal to M1 volume plus M2 dipole plus C, something like that. Okay, so here it can do multiple also. There is something called data analysis. I don't know whether you have used these statistical functions in Excel, something called data analysis. And then you can see, go down, down, down. You will see regression here. You see this regression, data analysis. You can do a lot of things. So here I give you the X range, Y range. So Y range is my activity. X, now I give both the X's combined together, volume and dipole. And then I will give you output range. So I will give some output range. Then I say, okay, so it's calculated. A lot of things, we look at each of them in detail. So it is done at 2 parameter linear regression. This is variable 1, that is volume, this is variable 2, dipole. And it is giving you R value, R square. There is something called adjusted R square. These are all statistics. As I said, I hope you have done a course on statistics, because statistics is very important for experimental studies and especially biology, and regression. QSAR uses a lot of statistics. So it gives you the intercept. So this is like activity equal to 0.0198 into volume plus 0.744 multiplied by dipole plus C minus 6.22. That will be the equation. And as expected, this is not a 2 parameter, is not a good model. That is why if you know p value, the p value is greater than 0.05. That means it's not significant. Okay, so here all the p values are greater than 0.05. So obviously, with so many limited number of data, it's not a good idea to work on 2 parameter, that is y equal to m1 x1 plus m2 x2 plus c type of models. If you have 8, 9, 10 data points, you can work on it. But Excel can do 2 parameter, 3 parameter, 4, any number of parameter linear regressions. Multiple linear regressions can be very nicely done with Excel. It gives you all the statistics to see how good the model is, what is the significance of the data. It gives you the range, 95% confidence limit for each of these values. It gives you standard errors. It gives you the ANOVA table, it gives you the F value, sum of squares of error, mean sum of squares, degrees of freedom, all those things it will give actually. Okay. Okay, so ideally, as you can see, regression has 2 degrees, residual has 2, total is 4, so there is no really extra degrees of freedom. That's why the model doesn't come out well actually. Okay, so like I've been saying, you need more data points to have a 2 parameter model, but Excel can do this also. Okay, now there is a Excel can do multiple regression equations also. Excel, we can even do second order, third order. Here we have done a y equal to mx plus c, but it can also do m1x square plus m2x plus c type of models also. I will show you that. Volume versus activity, for example. add a trend line. So you see, you can now add polynomial. That means you can also add okay, you can add second order equations also. That means y equal to a volume square plus b volume, like that also we can add actually. See, y equal to a into volume square plus b into volume plus c type of model. Already, even with the linear, it is very good model. So with the second order, we don't need to do a second order quadratic equation in this particular case, because already, as I showed you before, the model is very good with the linear equation itself. But you can do in Excel second order, third order, or whatever type of order models also can be done actually. So Excel can do a lot of multiple linear regressions and higher order models also. Okay. Okay, there is one software called Build QSAR that also can do multiple linear regressions. It almost looks like Excel actually. So the data I will copy. The data I will copy. This data I will copy, and I will show you how to use that. Let us see why. That is the activity here. From the Excel. So I copied everything. So this is volume, this is velocity, this is dipole. Okay. So we can look at the correlation. So choose the correlation between each of the indexes with the y. So like I showed you, know, volume is very good correlation. Dipole also is very good correlation. But this one is bad. Log P is bad. I've seen here. So we can do that correlation matrix. Then we can have covariance matrix. It's called covariance matrix. Then we can do. So we can plot each variable, that is volume versus activity, or log P versus activity. This is bad. Log P versus activity, or dipole versus activity. Or we can now develop a QSAR also. Multiple, this is called multiple linear regression. So here I am putting volume and dipole. So just like I got it in Excel, this also is showing the same thing actually. In Excel, we saw that, right? Yeah, same thing like Excel. This also will give you values point. Variable 1, variable 2, then it will also give you graphics. It will give you the experimental data versus what the model predicts. So all those things, then it gives a lot of statistical analysis. So it gives a lot of analysis, ANOVA table. Excel also gives you all those things actually. So this build QSAR can do the linear regression. It can do multiple linear regression. And then gives you the ANOVA table, p value, f value, mean sum of squares with error, gives you r square, adjusted r square, and q square. These are a lot of statistical terms. comes in regression actually. So this build QSAR can do multiple linear regression, just like the Excel which I showed you. Um, in Excel, you have that command data analysis, where you have a lot of data analysis facilities are there. There you have the regression. So you can do multiple linear regression. We build QSR also can do something called principal component analysis. Principal component analysis, again, it comes under data analysis and data manipulation. So you can combine X's, that is the independent variable, and come up with new variables which are called principal components. Actually, you must have heard about big data and data analytics and all that. So principal component analysis plays a very important role there actually. So it gives you principal component 1, principal component 2, and what is the loading of each one of them. As you can see, loading of each one of them. So three variables. So all the three variables or x's, it takes and converts it into three principal components, and we can use two of them and try to develop a model. So that's called principal component regression. It makes use of all the three variables, then comes up with new variable which is called PC1 and PC2, which is a combination of all these actually. So if you have many x's and you don't know which x to select, or you think many x's are going to be important, then you can use this type of principal component analysis technique to do that actually. So that cannot be done in Excel. The principal component analysis can be done in this build QSAR. So if there are too many x's and you think many x's are important, then you combine many x's into two or three principal components and use those principal components to develop your regression relation. So instead of x1, x2, x3, it will be like principal component 1, principal component 2, principal component 3, like that actually. This comes in concept of large data manipulation, data analytics. So different types of models we can do. I showed you now. Single linear regression model, either molecular volume or um, dipole moment, or multiple linear regression model. I showed you know molecular volume and um, dipole moment. But as I've been telling that it's not a good idea to have too many x values when you have only 5 data points. So 5 data points, always stick to one x value. Okay. Okay. One important point which I forgot to tell you is there is something called test and training set. Because you are going to use QSAR in predicting unknown molecules, it is not only enough to fit the data, but you should also be able to see whether the model you develop can predict other data. So what we do is, we whatever data you collect from literature to start your QSAR with the activity, we take only 80% of the data and we call it training set. The remaining 20% we don't use it. So with the 80% of data, you develop your QSAR, you got an equation, and then you try to predict the activity for the remaining 20% and see whether the model is able to predict. Understand? So there is the concept of training and test set, 80-20. So in our 5-parameter model, the 5-parameter model which we saw, it's not a good idea to use all the 5 data points and develop a model. It's wrong. So you should try 4 points as a training set, keep one of them as a test set. So you develop a regression with only 4 points, and with that model, try to predict the activity of the 5th point. And anyway, you know the activity, so you see how the model is predicting. That is the correct way of doing actually, the 80-20. Okay, that's very, very important. Yeah. So you divide it into training set and test set. Because QSAR is not only fitting data, but also predicting data. So how do you know it predicts? That is why you do training and test set. So if you go back to your original Excel problem, you should take only 4 points and get an equation. With that equation, try to predict for the 5th point you left out and see how good it is. Okay? So which one to leave out? You may have to try all of them. So that's what I did. What I did was, you have this 5. So I left out this. I used only the top 4. Okay, I used only the top 4. You can see only 4 data points are there. Okay, only 4 points I used. I did not use the 5th point. And then I developed the equation here, which will be different if you use all the 5 points. And then try to predict using this equation for the 5th point. So it is predicting 2.09, but actually it is 2.55. Okay, then what I did was, I removed this point but kept this point. So I removed 332, but I kept 420, this. So with those 4 points, I tried to predict, get a model. As you can see with those 4 points, I tried to get a model. And then I tried to predict the 332 point. Now you see, the model is predicting well. Model is predicting 1.84, actually is 1.89. So you need to do like that for all the 5 points, one after another. And finally, I got this, leaving out this point only. So this is a model which has got good fitting capability because R square is good, and it is also predicting well the data which was left out. That is the entire concept of training and test set. That is the entire concept of training and test set. So these are very important points in QSAR which one has to remember. Number one, 80-20, where you take only 80% of the data as training, rest of the 20 you park it and keep it as test set. And then if you have, for one parameter model, you should have at least 5 data points. Don't go for 2 parameter model with 5 data points. So that's a rule, actually, approximately 5 data points per parameter. So if you want to do y equal to mx1, m1x1 plus m2x2, have at least 10 data points. Okay. And then look at all the statistics which I showed, R square adjusted, Q square, F value, P value. From that, you will know how good the fitting is, what is the statistical significance of the fitting, all those things actually. Might have studied totally in regression analysis long time back. So the model which we have developed, mm, by leaving taking only 4 points, this model has good fitting as well as predicting. Whereas if I take this all the 5 points and develop a model, which I showed you originally, all the 5 points, we don't know how good it is going to predict actually. Okay, unless we have some other data where activity is known and we predict. So training and test analysis is very, very important. Anybody has any doubts so far? Any doubts? Anybody?
No, sir.
No? Okay. Continue with the Okay, let's look at another problem using QSAR. You can see this slide.
Yes, sir, it's visible.
Yeah. So imagine I collected some data, um, anti-inflammatory drugs actually. I got 5 compounds: licoxib, rofecoxib, valdecoxib, nimesulide, DUP 697, and the IC50 is given actually. Okay, they all experimental data which we collected from literature. Okay. They are anti-inflammatory drugs. We use some of them for our pharmacophore model also, right? Okay. Now I know the y value, so I have to collect the x's, or I have to calculate x's for each one of them, right? So first I get the silicoxib, yeah, now you must be familiar well with the smile and things like that, right? I hope you are familiar. So there is a good server which can calculate descriptors. Of course, even Swiss ADME also gives some descriptors, but this is a big list. It's called ChemDes. CHEMDES. Big calculates a lot of descriptors, almost 3,000 plus descriptors. Molecular descriptors. What will you derived properties? So basically, they are physical, chemical, topological, chemical molecular. It's a free web-based platform for calculation, so everybody can use it. 3,679 descriptors. Big number actually. A large number of descriptors you can calculate. Can you see the screen?
Sir, your screen is visible, but you are not audible.
Yeah, now?
Now you are audible, sir.
Okay. Okay. You can hear me now?
Yes, sir.
Okay. 3,679 descriptors. This software can do actually. And if you want to know more details about what each descriptor means, some descriptors here it's easy to understand, comprehend. Like we are all used to volume, surface area, log P, hydrogen bond donors, acceptors, like that. But there are many descriptors which come from graph theory, topology based theories, so which you will not be able to comprehend. So you can understand what this descriptor means. You can look at literature also. It gives you some molecular descriptor. So many descriptor libraries it gives, so we can get details of these details. So it tells you what more details of issues. There are connectivity descriptors, how the bonds, how the atoms are connected. It's called connectivity descriptors. More like a valence based descriptors. So like that, lot of information is given. It will give references also, so that if you really want to spend a lot of time understanding what this descriptor means, we can do some reading actually. Okay, so we can calculate these descriptors for each of the molecule. So you can input Smiles notation here. So you're all very familiar with Smiles. So the first molecule, for example, first molecule, for example, is silicoxib, right? Silicoxib. Okay. Celebrates, we got the smile, then input the smile notation. and then we say submit, calculates those descriptors. Yeah, so it's calculated some descriptors. Okay, then we can calculate other type of descriptors. These are called CDK descriptors. So again, these are called CDK descriptors. Then we say submit. Okay, so a lot, it's calculated many of them. Some log P and all is coming. Now you can download this data, download CSV file, download CSV file. Yeah, CSV file is Excel file. So the beauty is we can nicely take it to Excel and organize them. That's the advantage. CSV file. So we can calculate each one of these descriptors. You can try the attempt test now. All of you, you can go to Zinc, get the smiles of each one of the compound. Try it out and see. So you can do like that for each molecule, all the descriptors, and you will get Excel file. You can combine all the Excel files. So you should have around 3000 plus um, descriptors for this molecule. Correct? Just try test and see whether it works on your screen. Get the data from Zinc and then celebrates opinion of this molecule. Just try it out and see whether it works on your screen, on your laptop. Download the file, CSV, that will look like an Excel file.
Excuse me, sir.
Yeah. Were you all able to use ChemDes? Copy from Zinc, run ChemDes? All of you? Anybody
Yes,
having problem?
yes, participants.
Anybody has any problem? Are you able to take the structure, smiles, go to ChemDes, download, then
Yes,
CS,
sir. Yes, sir. They are able to do.
all of them?
Yes, sir. They are able to do. That's right.
So you have to do one after another, and for each one of the yes, then you have to assemble them all together. You downloaded um, for each one of the descriptor set, you will get a CSV file. Then you have to put all the CSV files together so that you will get approximately 3000 plus rows actually. Like that you have to do for each one of the compounds. So that's a little bit of work. You have to be thorough in Excel. I don't know how thorough you people will be actually in Excel. Um, okay, so what I did was so you have to go take one compound, go to zinc, get the smiles, go to ChemDesk, and then you run one descriptor set because ChemDesk is different descriptor sets. Then get the CSV file, go to your one master Excel sheet, copy that. Then you run the other descriptors, get that CSV file and copy that into the master. Like that, you have to keep doing for all the descriptors for compound one. Then do the same thing for compound 2. Do the same thing for compound 3, 4, 5. That way you will have a master file. This is the master file. Here I have the activity. That is my Y. And here I have all the descriptors. You can see. So you have to assemble this finally. It doesn't happen on its own. You have to do it yourself. Can you see the Excel sheet? Can you all see
Yes,
the?
sir.
Yeah.
Yes, sir.
So,
Yes.
so you have to assemble. So it's a lot of work actually. Lot of work. You see, I have put only 600 descriptors. But that's how you have to assemble. So each molecule, get the smiles, and then calculate all the one descriptor set. That is 1100, and then get the CSV file, copy it in the master file. Then for the same molecule, get this descriptor, and get the CSV, copy those results into the master file. Like that, you need to go through entire lot, then go to the second molecule. Second molecule means, say, first celebrates, then the other one is trophic oxide, like that. Okay, and then first row, you can put the activity values. Activity values is the experimental values. This one. You can see that, right? That's the Y value. Okay. So this will be your master file. All the descriptors down will be there, and Y is given here. Okay. Now I have to see which descriptor describes my Y well. So I can do a correlation, right? Correlation of Y versus this descriptor. Like that, I am doing correlation for all the descriptors down. Because this is 0, it's giving error. Otherwise, if there is some number, it will give you some number. So I am trying to see what is the correlation coefficient. There are some good ones, 0.92 and all that, but I want to select the best ones looking. So I go down. I think I have marked in red also. So this particular descriptor, BCUT P3, okay, and activity, you can see correlate with, and the activity, that is this one, as the 0.985 as a correlation. That's very good. Okay. So this could be a descriptor for my model. So y equal to mx plus c. The x could be this CUTV3 descriptor. Okay. So again, I keep going down. I get a lot of 0.72, 0.8, 0.91, but this so far this is the best, 0.985. So let us see whether I get something good. Down. 0.6, 0.92. This column. Nothing is close to 0.9. Everything 0.8, 0.85, 0.7, and all sorts of numbers are there. Mm. 0.99. Fantastic. That's a very good one. So between the activity and this particular NOxY, I need to find out what NOxY is. 0.99. I think this is the best. 0.998. There is a 0.94 also is there. Some 0.98, 94. So there are very good numbers are there. Okay, so I think this will be the best descriptor to select. This one. So this could be my X and activity could be my Y. I can get a Y equal to mx plus c like equation. I think I've drawn it also somewhere. I have not drawn that actually. I will take that. Yeah, I have taken this descriptor and drawn this descriptor BCUT3. Yeah, and this is the equation. It's coming out 1.209 BCUT minus 39.17. Good, very good. I didn't notice the other 1.998, so we will select that also.
Then one of the participants, Jaypal Singh, has something to say. He has raised his hand.
Ask him, what is the question? Yeah,
Just
go
a
ahead.
second, sir. I guess he has left the meeting.
But disconnected. Maybe when he comes back, please ask him.
Yes, sir. Yes, sir.
I've selected the other descriptor also, and just to see how the because correlation coefficient is very good for that descriptor. Yeah, this is the other one. Increasing of that decreases activity. Okay. Whereas in this other one, that increasing of that descriptor increases activity. In this one, increasing of that descriptor decreases. Oh, again, Oh, sorry. So X is that descriptor and Y is my activity. This is the activity. Okay. Okay. So in this particular case, it's going down actually. As the descriptor increases, activity goes down. So I should have less of that. Add trend line. Okay, so this is the best QSAR, better than this, but we can always consider both the QSAR. Both are good actually. So if you use this descriptor, you get the equation which is very good, 0.9711 up there. And if you use this descriptor, it's coming out 0.99. So here, as the descriptor increases, activity decreases. Extremely very good fit actually. So this is also because 0.99, 0.97 are not very far from each other. So we can consider one of them as our QSAR. So here we have a one parameter linear regression model to describe this particular thing. We can even we can see whether we can do a 0.9 over here. So I have also done a spire term, that is a quadratic term, but it doesn't improve further. 0.97 itself was good. This made it 0.98, so it's not a big deal. And this descriptor, it comes out very good for 0.99. You can't go beyond that actually. So Excel came this together and do very good multiple linear regression models. Okay. I don't know how, like I said, I don't know how familiar you are because the data analysis has the regression equation. So you can see regression. So we can nicely develop multiple linear regressions also. Like I showed you in the other case, it gives you a lot of statistics. Okay. Statistics are very important for us to understand how good the fitting is. Y range, for example, X range. I yeah. So this data analysis in Excel gives a lot of statistics, which is very important for you to understand. Okay. It gives you R square, adjusted R square, standard errors. Then it gives you an idea about the ANOVA table here, statistical significance of the fitting, and then it gives you range. All these are very important. So we need to understand that. So you people have to learn how to use this data analysis if you want to do QSAR. Many people don't do much of QSAR because it involves a lot of statistics and understanding of statistics. That's important. Okay, so you need to understand all these terms and see the importance of it. Any doubts? Anybody has any doubts?
Yes, participants. Any query? So no, sir. None of them.
Any doubts?
Sir, no, sir. They don't have any doubts, sir. Please continue.
So you are all familiar with the statistics, data analysis, and all? Are you all familiar with this ANOVA table and p values, lower and upper bounds? Anybody? All of you are familiar because this gives you all the statistics and you need to see how good the fitting of the data is. So we have come up with the two possible.
Excuse me, sir.
Yeah.
Sir, can you please a little bit louder? Participants, a little bit louder.
Yeah, we can use SPSS also, but SPSS you have to pay money, right? So SPSS you need to have. Yeah, but then if you have funding, you can buy any softwares you want actually. SPSS you may have to have annual renewal license and so on. So it depends on whatever SPSS gives, all the statistics this also gives. So the understanding of the statistics is more important than what is given also. So because SPSS also gives all this, but the understanding of the ANOVA table, understanding of the p values, f value, then range, 95% confidence, that's more important. It's exactly the same as what SPSS gives. See, my course has been always use of free software, but if you can afford any other software and you can pay annual license, then you can do that actually. But then Excel is one of the best software, well standard, um, and they keep on upgrading with a lot of statistical things actually. For example, if you go to R software, R is a free software and it can do a lot of things actually. So don't think that free software you need to buy an software which you have to pay for, which is better. That is not true, because nowadays many things are coming out as open source. The idea is to make things available for everybody and anybody, not just those who have money. Okay, so that concept is completely wrong. That's how it has been. But you see a lot of open source which I've been showing on molecular modeling and drug design. Many are coming, and this is what many researchers have realized, because the companies are trying to corner by having very high license fees. Some of the commercial softwares, you have to pay annual license fee, annual renewal fee, and especially country like India, 60-70% of the university colleges cannot afford them actually. So, and don't think these softwares are not very high end. They are very high end actually. You can publish papers with this software. We have published a lot of papers with these software.
Accuracy is
Accuracy is
policy.
yeah. See, if you go for a commercial software, the basic core algorithm is developed or bought from a professor in some university research, right? Then they make it to build up on top of it with all nice graphical user interface and all those things. So it is not a commercial company starts developing the basic core, that is basic mathematics part of it. That is maybe bought from a professor, researcher. Then you
It's
add
a
the you add the
something.
you add the graphical user interface, you add the input output systems, you add the workflow. That's all done by a commercial company. But the basic core, for example, if you take GRUMAS, if you take Autodoc, if you take Autodoc Vina, um, all the Swiss-based, Swiss model, Swiss, all those, they're all free and they all developed by top-notch people. Actually. The basic Grumax, Charm, they're all Mopac, Swiss-based software, Swiss modeling-based softwares. All the force fields like MM plus, MM2, MM3, all of them are developed. So the entire world is, researchers are going towards open source. That's how everybody. R is a good statistical software which is freely available. You have to learn it to use it actually. It's
Like
freely
people
available.
go for this paid software Schrodinger that it give very accuracy. So,
Nothing like that. Because if you go for a commercial software, you have to pay annual license, right? Yeah, like 6 lakhs, 7 lakhs. If you can afford it, it's okay. But then otherwise, it's difficult. And the open source softwares are also developed by researchers. So all
yes,
softwares are developed by researchers, not some computer scientists. They're all different. It's developed by biophysicists, correct? Biophysics experts or not by computer scientists, because computer scientists can do only graphical interface. But the calculations, force fields, they are all have to be developed by biochemists and biophysicists. So any software, and even paid software will always go through the basic algorithms of semi-empirical calculations, quantum mechanic calculations, which have been there for a long time. And all softwares will give a lot of information, but the idea is you have to understand each one of these terms and try to make sense out of it actually. Whether it is if you go to regression software, there are so many. Minitab is there, SPSS is there, GraphPad is there, Prism is there, so many softwares are there. They'll give a lot of statistical information, but if you don't understand it, is like useless. You should be able to understand and make sense and develop your work based on that. That's how it works. So for QSAR, you need to understand statistics. That's very, very important. You need to understand statistics to see how good the modeling is actually. So as a chemist, I don't know whether you people do any, especially researchers, maybe in research methodology, they do some statistics, right? Or do they do statistics in research methodology in PhD scholars?
so like mostly like bio people they don't have mathematics background. So is it
No, no.
convenient for them to?
In PhD, everybody has to do a course called research methodology. In that course, they are taught statistics. All universities, all everywhere. I think it's become mandatory. I don't know what
Yes,
colleges.
nowadays it is becoming mandatory.
Yeah, it's mandatory. Anybody who is doing research has to undergo a course called research methodology, and statistics is taught there actually. All these terms have to be taught because, especially for data analysis. Maybe chemistry, you are not generating data, so you don't analyze data, but if you are doing biology, if you are doing engineering, if you are generating data in engineering, generating data in biology, we need to do that actually. Yeah, bioinformatics, lot of data analysis. So all these statistics will come there. Bioinformatics, you are doing bioinformatics, data analysis, sequence comparison, lot of statistical terms. But pure organic synthetic chemist, I think, will not be handling data much actually. Right? We don't come across statistical terms, terminologies. Yes, like problem is there. But there is a lot of mathematics in this course, which I am not covering. It's not difficult mathematics, but a lot of mathematics is there.
Yeah, that molecular mechanics part.
Yes, molecular mechanics, force field, optimization is there. So I'm not covering all those. We are just showing how to use software. But then when you say computer aided, you have to have some mathematics, right? Otherwise, how will computer come into the picture?
Yes,
So,
sir. Yes.
so it is a lot of mathematics. So minimum energy conformation is there. Any molecule takes up a minimum energy so that the binding the energy is minimum. That is the most thermodynamically stable, right? So the molecule attains that particular energy. So there a lot of force field, a lot of optimization comes in. A lot of optimization comes in. So I will show you that now.
That part is required in docking also.
That part is required if your molecule is not there in zinc database. For pharmacophore, suppose I have one new molecule I have synthesized in my lab and studied the biology. It is not there in zinc database. What will you do? So I need to get the confirmation of the molecule, right? So I have to use a software called Marvin Sketch. That's a free software. And so it calculates, it adjusts the shape of the molecule, bond angle, bond length, so that the energy, the internal energy of the molecule is minimum, because thermodynamically any system attains a minimum energy, right? So there is a software. It's freely available for teachers. It's called Marvin. And did you all download this? It takes 3-4
Sir,
days.
participants, yes, sir, participants were saying that they downloaded, but it was asking for a license key.
You have to write to them using, I mentioned, right? You have to write to them using your institute email ID, then you will be given a license. If you write Gmail ID, they will not give. You should give
Okay.
institute email ID. This is a very good
Some organizations they don't have.
institute. If I am a college, then college will have some institute ID, right?
Okay, institute mail ID,
That's what I mean. Institute email ID.
but call it domain, sir. Institute mail ID, that is Gmail ID.
No,
Call
Gmail
it domain ID. Okay.
because they don't want to give it free for everybody. They have a paid also. They have a free for academic people. Okay, so like you and me, academic people. So they want to give it free. And this is a very excellent software. It can do the minimum energy of the molecule. So imagine I don't have a structure uh, available in zinc. Then what do I do? I need to get the three-dimensional minimum energy, because that is the conformation any molecule takes ultimately. Thermodynamically, that's what it is. So I will draw that structure. So all of you get this using your institute ID. It will take 4-5 days for them to send you their key. So I'm just giving some structure. imagine I have drawn this. So the structure is unfortunately not there in sync database. I've drawn it on a paper, so it's like a flat molecule, but it will assume some three-dimensional conformation, right? All of you know that for a minimum energy. So we can use this Marvin sketch. First we draw it on a paper. This is like a flat, but actually in real life it may take a three-dimensional confirmation, right? So structure, we say clean in 3D. So we can do a lot of things with this. We can put wireframe, ball and stick, so many things it can be done. We can go through it. We'll see implicit hydrogen, explicit hydrogen, space filling modules. Yeah. So many things it can do. Okay, now I want to get the minimum energy conservation of this molecule. So I do so. There is something called calculations, confirmations. So this also can give a lot of descriptors. See, solubility, many descriptors. We'll come to that actually later. It can also give a prediction for NMR prediction also. So it's a new molecule. So NMR is not there, but it can give you NMR prediction also. Okay, confirmations. Okay, so see force field, and so a lot of mathematics is inside actually, which I am not talking about. So it will adjust the bond angle, bond length, and torsion angle. Torsion is like a three-dimensional, third dimension, and they give energy. See, 51.27 kilocalories. So 51.63, and so you can see. So this particular confirmation is the, you can see this. This is the minimum most energy confirmation of that molecule. Energy is this. So I can copy this, file save, um, save as or save selection. So many things are there. We can save it actually. So we can save it. And then if I am doing docking or if I am doing pharmacophore modeling, because pharmacophore, what it does, it superimposes many structures, right? Superimposes many structures. So that way it's very, very useful. This is the minimum energy of that molecule. This is the minimum energy confirmation of the molecule. And this is the minimum energy of the molecule, actually. And this also can do a lot of tools. Yeah, in tools, it can calculate many descriptors like polar surface area, molecular surface area, log P, so many things it can calculate. Yeah, this is the polar surface area of this complex. You can see the screen, right?
Yes, sir, it's visible.
Yeah, okay. So lot of other calculations, descriptor calculations can be done. You can look at charge, polarizability. Um, yeah, so it gives you the charge distribution of the molecule. So negative oxygen is there, no? So you can see the negative here, red color, blue is carbon. So it gives you charge distribution of the molecule. Numbers also are given here. Charge localization. Then it can do geometrical descriptors. It can give all the other details. So this also see a lot of values it can give. Yeah, lot of numbers. See, energy, projection area, van der Waal volume. So many descriptors also it can give actually. Steric hindrance. Then it can give pKa value. It can give log P. This is a very good software. All of you please get it and keep using it. As a chemist, we can draw new structures. Log P. This is based on group contribution method. So oxygen is more hydrophilic, so more negative. All carbons are hydrophobic, so positive. So this is called group contribution based method. Log P is 3.26. It's very hydrophobic molecule, right? A lot of aromatic groups and benzene. CH2 groups only few oxygens, three oxygens. Okay, so this is a very excellent Marvin software. Once you get it, you have to play around with that. No, that's very important. All the commands go through. Whatever. If you are a research scholar, you can draw your research synthetic molecules, get the energies, then get some parameters. Then, if you are doing some derivatization, draw them, and again get minimum energy confirmations, get various parameters, study each one of them. That's very important actually. It gives you a lot of molecular properties. Yeah. And pH donor acceptor at different pH conditions, how the number of donors acceptors change, especially if it's going to get protonated, then that number will change, right? I think it's nothing. Yeah. At this pH, I think it's getting protonated. So this is an excellent one. Did anybody get the license?
Sir, actually, some participants are having problem of that mail ID only. They are not getting. So this is their problem.
They don't have a domain ID,
Yes, yes.
institute ID. So they should ask the. What about their teachers?
Okay.
They can contact other person.
Other person? No. Forget about students. What about the teachers may have some domain ID?
So,
Then they can
so
teach,
that's what I'm saying. Like
even colleges.
some colleges, they don't have their domain ID.
Oh, okay, okay. Then
And
they
because
have
nowadays, I think it is chargeable.
yeah, correct. It is chargeable. Yes, yes, yes. Then they
It
have
is
to.
not. Nowadays, it is chargeable. And many teachers, some are on contractual basis, so they don't have
Oh,
at present. That.
oh, okay, okay. Then they may have to ask their colleagues or something, right?
Yes. Yes.
And if some teacher gets it, they can give it to their research scholars also, right? For them to study some of these their own synthetic molecules.
Yes.
Of course, this also has a paid version, which will have more facilities, but this is a free version for all of us. So getting a free version is very useful, because if you are going to do
This is a very good software. Marvin Sketch is a very good software.
yeah, one of the best actually.
It's a very good, but the only problem is that regarding mail ID.
Yeah, they have to talk to somebody else and get it actually. Because if you are having new molecule and you don't have the SDF file from zinc, if you want to do docking, you don't have a mol2 file available anywhere because it's a new molecule which you have designed. Okay, if it's an old molecule from literature or if it is a drug, it will all be there somewhere. We do a search, Google search or some scholar search, you will get it. But if it's totally new, which you are designing, then it will not be there. So definitely you want to see the structure, you want to see the properties, and then you want to derivatize that molecule, then again see how properties change. So for all these, nothing like having a Marvin. Nothing like Marvin. You can do so many things. You get the three-dimensional structure, and this is a very flat molecule, so it does not take in because you have two benzene rings connected bond, so it's a very flat. You can see this, right? Very little bit bent between this benzene and this benzene. Otherwise, basically, it's very flat structure. But you may have molecules where there could be some intramolecular hydrogen bonding also. So those things and all we can study nicely. If it really gets twisted, or if it is getting completely helical in shape, then intramolecular hydrogen bonds and all you can study. Then you can calculate some properties of this molecule. As I showed you, it can even do pKa value. And this molecule does not have much.
So if our colleagues are working in other universities, so we can take from them.
Yeah, yeah, you can take from them also. If they are willing to give, you can take from them. It can give you pKa value also, so that's good actually. pKa. And that way it will be very useful for chemists. According, that's what I think. Even synthetic chemists who are designing new molecules, wanting to have a look at how the structures are, how the molecule conformation changes if you are putting in some new derivatives, right?
Yes.
New functional groups on the sides, all those things.
So we have to take the minimum energy conformer.
Yes. So you can look at how it changes. So Marvin Sketch is such a software for that, and this uses force field. There are software which uses quantum mechanics also, semi-empirical quantum mechanics. I'm not teaching that. That is a software called MOPAC. That's also free actually. Good ones are always free. Okay, there is one TV advertisement also. You must have seen, no? All good things are free. Yeah.
Yes.
So, the MOPAC is an excellent semi-empirical quantum mechanics software. One of the oldest software systems. that actually, because Marvin calculates the information minimum energy based on force field, whereas MOPAC calculates based on semi-empirical quantum mechanic theory. Okay, so there is a difference. Yeah, this is MOPAC. It's very old actually. One of the standards for semi-empirical, I would say almost 30, 40 years old. Okay, it calculates semi-empirical quantum mechanic energy of molecules given a structure. It calculates the highest occupied molecular orbital, lowest unoccupied. It will calculate charge distribution, all using semi-empirical, not full quantum mechanics. Because, as you know, full quantum mechanics, big molecules, you can't do much. Even 50, 60 atoms means full quantum mechanics, you can't do anything. Whereas the semi-empirical is an approximate method, but it can do large atoms, molecule with large atoms. Whereas force field cannot give us highest occupied molecular orbital, lowest occupied, the energy required for jump, shifting from lowest occupied to highest, all those things, because force field which Marvin uses does not consider the electron and the nucleus. It is considering the entire electron and the nucleus as a single ball. Whereas in quantum mechanics, the electrons are different, nucleus that which contains protons, neutrons are different. So you have those proton-proton repulsion, proton-electron attraction, electron-electron repulsion, all those things are there. But semi-empirical makes it more simple because it's called semi-empirical. It's much more simpler. So if you want to calculate things like energy required for moving from one orbital to another, energy required to break a bond, energy required to have a charge on a molecule, H plus dissociation, you have to use quantum mechanics and semi-empirical quantum mechanics. This is MOPAC does that actually. And so it gives you all the information related to that actually. This is also free downloads. Okay, so you can download the MOPAC. There is an instruction manual. So if somebody is interested in looking at those type of electronic details, then you go to it. If you are not interested,
out of these two, we have to download standalone 64-bit MOPAC 2016 for Windows.
For Windows, yeah, for Windows, download standalone, yeah.
Standalone.
Yeah, I have got it actually. So you can download. It's very simple actually. And if you are interested in like electronic features, electronic energies, orbital energies, force field, Marvin cannot get those details. You need to use this semi-empirical MOPAC. Okay. If you are interested in that aspect, if you're not interested, then doesn't matter. You can just use Marvin. But if you are looking at electronic energies, energy required for moving from one orbital to another, ionization potentials, so such calculations, if you are interested, then you need a MOPAC actually. Okay. Otherwise, for minimum energy calculation of a molecule, if I am going to do pharmacophore models, if I am going to do docking with ligand protein, for such things, actually, Marvin is enough. Okay.
So in some cases, the protein molecule is very big. So
you can use semi-empirical quantum mechanics. So nowadays, softwares are coming. Only the active site they use semi-empirical quantum mechanics. Rest of the protein is force field. So softwares are there with the combinations. We can do that actually. Because protein will have say 100 kDa molecular weight. If you do semi-empirical, you can't do semi-empirical for all the 100 kDa. Nothing will run. So only the active site portion, maybe 30, 40 amino acids alone, you do semi-empirical quantum, rest all will be simple force field based approach. Where in the force field based approach, the electron, proton, everything is combined as one single ball. So you have balls connected with springs. So the bonds are assumed as springs and one atom is considered as one single ball. So it's not differentiating the electron and the proton. That is what is the force field method. That's what Marvin does actually. It is good for all our docking because like I said, no, protein will be some 100 kilodalton, huge. You can't do anything with the semi-empirical quantum mechanics.
So this part will be discussed in docking.
No, I'm not going to show about semi-empirical quantum mechanics.
Okay.
I'm not. There won't be any time as such, right?
Right.
Okay. So anybody has doubts on force fields? Anybody, any doubt?
No, sir, till now there is no doubt. Yes, participant, any doubt or any query?
Any query, any doubt? So this is how the workflow was on QSAR, right? We went to Zeek database and we got the Smiles notation. We went to ChemDes maybe and then got all our descriptors. We use Excel, we use build the QSAR or any such thing, and we get that good the QSAR model. We divide the data set into training and test set so that the only training is used for developing your model. Then if the structure is not there in your Zinc, you have to use Marvin. Draw the structure and get the three-dimensional configuration. I showed it very fast, but you need to, if you want, so you have a Marvin, it will be good for you to practice on that. Actually. Marvin has a lot of commands, so you can play around with that. So that is the workflow in QSAR. Some points to remember, which I mentioned. With 5 data points, develop only one descriptor, like this. Don't try to and all that. Only if you have about 10 data points. Then divide the data, whatever data you took from literature, as a training and test set. Training 80%, test. I showed you when we had 5 data points. Now you should take 4 as your training and 1 as your test set. Don't forget that. So you fit the model only with 4 data points and then try to predict for the left out point and see how good it is predicting. So if you have 10 data points, fit a model with 8 data points. That is why this point comes in, it's 5 data points. 5 data points means it's only 4 data points actually, right? Because one is used as a test set. So with the 10 data points, it's like 8 data points. That's why these 2 rules are very important. And then you look at all the statistics. That's very important. Excel add-on gives you that regression gives you R square adjusted, R square predicted, Q square. All these have to be quite high. As I said, maximum is 1. This will be larger than this. This will be larger than this. Like that, it will be. So the numbers have to be quite high, tending towards 1. It will not be 1. It will not be even 0.9 because in experimental, always there will be experimental errors. So those errors also will come into the picture actually. Okay. So this is the entire workflow for your QSAR type of model, right? No doubts.
Yes.
So please, you have to learn some of the statistics if you want to see how good your model prediction is. Actually, you can always click buttons and get some QSAR, but that is not correct. You have to see the fitting. You have to see the predictive capability. You have to see the significance. All those aspects have to be looked at. Okay. You use that model later on for predicting some new designs. You are designing some new molecules in your bench before going to lab. You can always check out the predict the activity and come back. So this is good. This is not very good, and so on. Okay. Any doubts so far?
Fine, sir.
Participants, any doubt?
Sir, I don't think participants any doubt? No, sir, they don't have.
Anybody has any doubt? Okay, no, sir. Because I want to get some feedback on any doubts. That's important. Okay. Okay, the next part so far we looked quite a lot on the ligand based. I'm not going to talk about this. This is not used much. Only these two are very important to use. So we looked at the left part, which is called ligand based, where you have the lead molecule and trying to design new lead molecules. You have no idea about the target protein. Okay, that is why this is called ligand based. This side, the right hand side, is called target based, where you have some idea about your target protein. You know your drug is going to bind to this particular target. So I have some idea. I have some idea about the target protein for enzyme. I make use of that knowledge and see how my small molecule is going to go and bind to the target. That is what is called the target-based approach. So that means I need to know the crystal structure of the target. That's very important. Okay, that means generally X-ray crystallography. One is a two-dimensional sequence may be available. Sequence is like amino acid sequence. There are millions of proteins whose amino acid sequence are known, but there are only thousands of proteins whose three-dimensional structures are known. Millions of proteins, two-dimensional sequence are known. That is easier. I can purify, and there are sequence analyzers are there. But if you want to get a 3D structure, I need to crystallize the protein and have a X-ray crystallography done and get the 3D structure, which is very time consuming. Purification steps are there, so many things are there. So there are very few proteins on three-dimensional structure, whereas there are large number of sequences of proteins are available. Okay, so what do we do? So I want to have a protein for human which is involved in inflammation, COX-2, cyclooxygenase 2 enzyme. I may get it from rat or mouse or some other species. So I do something called a homology modeling. I make use of that 3D structure and I build the structure of my human protein. Okay, that's called homology modeling. Because I might not be able to get lot of protein purified from human source, whereas I may be able to get the protein purified from other sources, right? Rats or mouse. So that's the problem actually. So if I suppose I want to get a protein on some cancer, getting it from the human source may be difficult because I need enough pure. Generally, it will not be pure. It will be very impure. There will be 200, 300 other proteins. So I have to isolate the pure protein in sufficient quantity. From human, it may be difficult, but it may be available from animals because there could be animal studies done, genetically modified animals, all those things. So I may have generally for a lot of animals many different proteins related to our diseases, but the corresponding human protein might not be available. So the crystal structure might not be available for human, but it may be corresponding structure for rat or mouse or rabbit or something. So I do, based on the 3D structure of the animal, I build a 3D structure for the human protein. That's called a homology model. Okay, then I dock my ligands or drugs to that. Okay, that is what is called homology. This is going to happen in many cases. Like I said, there are lakhs and lakhs of sequences of proteins available in a database, but there are only 10,000 3D structures of protein available in the another database, because getting the protein in pure protein in sufficient quantity, purifying the protein, crystallizing the protein, and then running X-ray crystallographic setup to get the structure, 3D structure, is a very tough task. Um, so there are databases for 2D sequence. There are databases for 3-dimensional structure. I'll show you those two databases. So one is called UniProt. UniProt. This is a database which gives you sequence of lot of proteins, lots and lots of proteins. Okay, so if I give the UniProt number, I can get the sequence of that protein. Lots and lots. So suppose you researcher is isolating a novel protein, getting the sequence. If it is not there, you can upload that in the UniProt. See, look at this on the right. Can you see this page?
Yes, sir. Yes, sir.
Yeah, you on the right hand side, if you see two SARS-CoV-2 coronavirus protein entries. So
Yes,
they
sir.
have added the sequence of two coronavirus proteins recently, actually. So it contains a lot of sequences. For example, this prostaglandin synthase is involved in inflammation. So imagine I am interested to see, this is the sequence ID of that protein. Okay, just like we have our institute ID for each one of us. So we have a sequence ID. Okay, so this is a mouse sequence of the enzyme called the prostaglandin GH synthase 2. It's also called a cyclooxygenase 2. So it converts arachidonic acid. It's an oxidoreductase enzymatic. So it's got two activities, oxidation then reduction. So it converts arachidonic acid into PGH2. So it's involved in inflammatory pathway actually. So this protein sequence is there. This is the UniProt number. They call it, or it's also called Swiss prot. Okay, so you will hear about Swiss everywhere. We will do Swiss later also. So a lot of information is given about this particular enzyme. Lot of information will be given. Okay, so its function is also given actually. Lot of information. Yeah, details about binding site, active site, where the and then what is its function. Lot of information is given. It's from mouse. And this is another protein which is involved in asthma. This is the sequence. This is called 5-lipoxygenase. 5-lipoxygenase. This is involved in asthma. So how it works also is given. A lot of information also is given about this particular references. So many things are given. So this UniProt is a very useful database, huge number of proteins, and but it gives you only sequences. If you want a 3-dimensional structure, then there are a database called PDB, Protein Data Bank, Protein Database. Yeah, you can see this.
Yes, sir.
Yeah. So this gives you 3-dimensional structures of proteins that are saved. Like I said, you will see maybe 10,000s in that number. Whereas if you look at UniProt sequence, you will see lakhs and lakhs because, like I said, crystallization and all is a big problem. Okay. Okay, here also we can search a protein and see this is a 7RR0 SARS-CoV-2 receptor binding domain bound. The latest entry. So there must be a lot of research going on SARS protein targets. So this is the protein. It's called 7RR. Let's see. Let us see what its function is. So this is from a Homo sapien viral protein immune system. So experimental data, very recent actually, 6th of October, 2 days back. So they have used electron microscopy to get the structure. NMR is also used to get structure. The resolution is not fantastic, 3.1 angstrom only. But if you do X-ray crystallography, you can go down to 1.5 angstrom also. But the people want to do a lot of work quickly on SARS, so they come up with lots of this. Okay. 1pxx and this is the crystal structure of diclofenac. Diclofenac is used for inflammation. It's bound to cyclooxygenase 2 enzyme, which is involved in the inflammatory path. Okay, this is the enzyme. You can look at the 3D structure, cyclooxygen, cyclophenol can be seen here. Yeah. And you can see diclofenac. This is the enzyme cyclooxygenase 2. It's got the two. It's I think dimer. So it's got two. Here it is, diclofenac. Can you see it? Can you all see this? Diclofenac.
Yes, sir. Yes, sir.
It's in a ball and stick model. Down, right down the screen. Because ligand is in ball and stick, polymer is in protein form. You see that, right? So generally, they try to crystallize proteins with some ligand bound because if you don't do that, the active site will collapse when you crystallize. So later on, when you want to do docking, the active site will be not really the real because it might have collapsed. So sometimes when you download same protein, cyclooxygenase 2, there will be a lot of proteins, different people, different organisms, different. I will show you that. Yeah, that's two. So different ligands, different environment, and so on. Actually, so you have to select properly for your study. So many cyclooxygenase 2 is there. There is a I given a search as COX2, cyclooxygenase 2. So you can see 6NL solution human have modified post-translational modified cyclooxygenase 2 like that, so many different same protein, different groups would have done different modifications to protein. They would have crystallized with different ligands also. For your if you're doing docking, you have to select the correct compound. Some other compound 5SC75 bound to COX2, something else bound to COX2, like that. You will have the different types of silic oxide bound to COX2. You remember silic oxide, which we have been studying for the past 2 days. Yeah, silic oxide. This is a commercial drug manufactured by Pfizer. So that's bound to COX2. So like that, you say a lot of same protein in different. Yeah, this is silicoxyl bound to COX. I think this is silicoxyl. Ligand will be ball and stick model. Here it is. Yeah, you can see silicoxyl. Can you see it? Yeah.
Yes, sir.
Yeah. So if you want to do similar studies, I think this protein is good for you to download rather than any other COX2. Although there are many COX2s, this will be more relevant. If you want to design similar molecules. Understood? So you need to plan accordingly which one to use for your docking. So please play with these two databases. They give a lot of information. The Unicode. So there are many constituents here in the entries. Different molecules don't constitute. Spike protein, coronavirus spike protein. It looks like a cloud, actually, three-headed cloud. And so look at this electron microscopic picture of spike protein. Then it looks like a cloud with three heads. Spike protein. Bioelectron microscope. For science, so this is not a human. Similar, many. For SARS-CoV-2 spike, like this, no, it's like a cloud. MERS COVID, Middle East respiratory syndrome. That was another disease, viral disease, right? You all remember that MERS? Anybody remembers MERS? It came about a few years back. Anybody remembers MERS?
Pardon, sir?
Anybody remembers MERS? MERS, Middle East respiratory syndrome virus, which
Yes, sir,
happened
yes.
a few years back. So
Yes.
the structure of that MERS is given here, actually. So basically, spike proteins look like this. Actually, it's a cloud-like with three heads. Cryo-electron infectious bronchital coronavirus. Okay, these are all beta coronaviruses. Yeah, this is another structure. Neutralizing antibody. So depending upon again MERS. So depending upon the problem which you are trying to solve.
Sir, there is one query from the participant.
Yeah,
That
we can download the entire protein and then delete the domains. So some proteins will have dimer, trimer, tetramer. So we can delete whichever has the active site and use it for docking. Yeah, that can be done. Okay. So because most of the proteins, none of them will be a monomer. There will be dimers, trimers, tetramers. So doing docking with such big proteins also is a problem actually. So we take it as a monomer and work on it. Yes, it can be done.
is asking any specific software is required to do so.
Yeah, I will show you. That's also a free software. You can download it. It's called UCSF Chimera. So we can say download file. Okay, so we have to download in PDB format. Okay, once you download in PDB format, uh, 6VV5. This is the protein. There is something called UCSF Chimera. You can get that free. UCSF Chimera. That's also free. Yeah, so you got the protein. There's a spike protein. Like I said, it looks like a cloud, you know. 3 heads actually. So this is a free software. You can download it. You can start learning each of these commands. So when I do this, it does a lot of things to the protein. Okay, then you can save it, make it smaller. Okay, so you can. This does not have many things. So this command is there, which is called tools. Um, sorry, structure editing. Tools are there, which can do structure editing is there. Uh,
So can we share the feedback link?
structure binding on it, structure binding analysis there. So you can then go to that dock trip. So it prepares the protein for docking. So it will delete, if the protein is big, it will take one single uh, unit, it will remove solvent, it will add hydrogen, all those things it will do. Delete the non solvent charge, add hydrogen, add charges, and then it will say, it will give you that. That you can use it for your docking, because many docking software you can't put such huge proteins also. Okay, many big proteins docking softwares will go not be able to work on. So this is called dock trip. Okay, so this, please download this also, UCSF Chimera, do all your work. Okay, okay, we will continue tomorrow on the
Yes.
docking part of it. Okay, so this is also a very useful one, UCSF Chimera.
Thank you so much, sir. Thank you.
Yeah.
Neha, please share the feedback link for today.
Yes, ma'am.
You also use Pymol, but this is the best actually. I would prefer this. This is a chimera rather than this, because it's meant for large proteins. Pymol, Pymol also can be used. Tomorrow I will show you. And also can look at protein ligand docking. This is a very good one for protein preparations. Both are free. Pymol is also free. All good things in life come free.
We will continue with the docking part from tomorrow. And
Okay, tomorrow.
thank you. Thank you so much.
Thanks a lot, sir.
Participants' feedback link has been shared with you.